मित्रांनो, आज आपण आठवी मधला जनरल सायन्स या विषयामधला चॅप्टर 1 लिव्हिंग वर्ल्ड अँड क्लासिफिकेशन ऑफ मायक्रोब्स हा वाचणार आहोत. तर वाचूया. चॅप्टर 1 लिव्हिंग वर्ल्ड अँड क्लासिफिकेशन ऑफ मायक्रोब्स. Can you recall? 1. What is the hierarchy of classification of living organism? 2. Who invented binomial system of nomenclature? 3. Which levels of hierarchy are considered while writing the name of binomial nomenclature? Biodiversity and need of classification. Last year, we have learned that all the living organisms on Earth have updated according to geographic regions, food injections, defense, etc. While adapting, many differences are observed in the organism of some species too. According to 2011 census, around 87 million species of living organisms are found on the earth, including land and sea. To study such a vast number, it was ensured to divide them into groups. So groups and subgroups were created considering the similarities and differences among the living organisms. In history, Carl Linnaeus in 1735 divided living organisms or living world in two kingdoms, Vegetalia and Animalia. Haeckel in 1866 considered three kingdoms, Protista, plants and animals. In 1925, Catton created two groups, prokaryotic and eukaryotic. In 1938, Complan divided living organisms into four kingdoms, Monera, Protista, plants and animals. This process of dividing living organisms into groups and subgroups is called binomial classification. Robert Hardy, Whitaker, 1920 to 1980, was an American ecologist. In 1969, he divided living organisms into 5 groups. For this classification, consider following criteria. 1. Complexity of cell structure, prokaryotic and eukaryotic. 2. Complexity of organisms, unicellular or multicellular. 3. Mode of nutrition, plants, heterotrophic, photosynthesis. Fungi, saprophytic, absorption from dead organisms. Animals, heterotrophic and injective. 4. Lifestyle, plants, procedure, animal, consumer, fungi, decomposer. 5. Phylogenetic relationship, prokaryotic to eukaryotic, unicellular to multicellular. Living organism, prokaryotic, eukaryotic, unicellular, kingdom 1, eukaryotic, unicellular, multicellular, unicellular, kingdom 2, protista, multicellular, kingdom 3, fungi, cell wall present but organisms can't perform photosynthesis, plant type, cell wall present and organism can perform photosynthesis, kingdom 5, animalia, no cell wall. Kingdom 1, Monera, activity. Take a small drop of curd or buttermilk on a clean glass slide. Dilute it with a little water carefully. Keep a cover slip. Observe it under high power of compound microscope. What did you see? Moving small rod-like microbes are lactobacillus All types of bacteria and blue-green algae are included in the kingdom Monera. Characteristics 1. All the organisms are unicellular. 2. They are made autotrophic or heterotrophic. 3. These are prokaryotic cells without distinct nucleus or cell organelles. Kingdom 2. Protista. Activity. Prepare a temporary mount of a drop of pond water on a glass slide. Observe it under low power and high power of microscope. You will find some motile microbes with irregular shape. These are amoeba. Characteristics 1. Protista are single-celled organism with well-defined nucleus enclosed in a nuclear membrane. 2. They have pseudopodia. They have pseudopodia or hair-like cilia or whip-like phageria. For locomotion. 3. Autotrophics. Example: Almrena, Bollworm, contain chloroplast. Heterotroph. Example: Amoeba, Paramecium. Kingdom 3. Fungi. Activity. Take a moist piece of bread or bhakri and keep in a container with lid for 2 to 3 days. After 2-3 days, a fine cotton thread-like stuff is found growing on the surface of the bread. Prepare a temporary amount of free thread from this culture and observe it under the microscope. Institutional work. National Institute of Virology, Pune is involved in researches on viruses. This institute has been founded in 1952 under the jurisdiction of Indian Journal of Medical Research. Characteristics 1. These are non-green eukaryotic heterotrophic organisms. 2. Most of are saprotroph. They feed upon decaying organic matter. 3. Their cell wall is made up of true and complex sugar called chitin. 4. Some fungi are thread-like and many nucleus are present in the cytoplasma. 5. Example: Bhakri's yeast, Aspergillus, Fungi-on-Cron, Panicillium mushroom. Through many systems of classification are introduced after the Whitaker, his 5 kingdom system is widely accepted. Use your brain power. Explain matters and dematters of Whitaker classification. Classification of microbes. Among the living organisms, microorganisms are largest in number, hence they are classified as follows. Microorganism: prokaryotic, eukaryotic. Prokaryotic bacteria, eukaryotic protista, fungi, algae. With reference to size of microbes, remember 1 meter is equal to 10 raised to 6 micrometer. 1 meter is equal to 10 raised to 9 nanometer. Bacteria size 1 micrometer to 10 micrometer. 1. Unicellular ellipted plastic organisms. Sometimes many bacteria together from colonies. 2. Bacterial cell is prokaryotic with cell wall, but distant knuckles or cell organelles are absent. 3. They reproduce by simple binary function. 4. In favorable condition, bacteria grow vigorously and can be double their number in 20 minutes. Protozoa size approximately 200 micrometer. 1. Protozoa are found in soil, fresh water and seawater. Some are found in the body of other organisms are pathogenic. 2. These are unicellular organisms with eukaryotic cells. 3. There is great variation in cell structure, organs of locomotion and modes of nutrition among protozoans. 4. These organisms reproduce by simple cell division. Example: amoeba, perimation, free living in dirty water, Entamoeba, histolica, cases, Amoebiasis, Plasmodium, Wick-wax, cases malaria, Algena, autotrophic, fungi. 3. Fungi size approximately 10 micrometer to 100 micrometer. 1. These are found on decaying organic matter and dead bodies of plants and animals. 2. These are eukaryotic organisms. Some are unicellular and others are visible to naked eyes. Saprotrophic observe their food from decaying organism matter. 4. They respond sexually and asexually by cell division or by bending. Example: baker's egg, Candia mushroom. 4. Algae size approximately 10 micrometer to 100 micrometer. 1. They are aquatic to eukaryotic unicellular autotrophic organisms. 2. Photosynthesis is carried out with the help of chloroplast present in the cell. Example: chlorella, chlamydomonas. Very few species of algae are unicellular, most of them multicellular and visible with natives. Virus size approximately 10 nanometer to 100 nanometer. Generally, viruses are not considered as living organisms, or they are said to be organisms at the age of living and non-living. They are studied under microbiology. 1. Viruses are extremely minute. Example: extremely minute, i.e., they are 10 to 100 times smaller than bacteria and can be seen only with electron microscope. 2. They are founded in the form of independent particles. Virus is a long molecule of DNA or an RNA covered by a protein coat. 3. Virus survives only in living plant or animal cells and produce their own proteins with help of host cells and create their numerous replica. Then they destroy the host cell and become free. These free viruses again infect new cell. 4. Viruses cause many diseases to plants and animals. Do you know human poliovirus, influenza virus, HIV, AIDS virus, etc. Cat leg, P. coronavirus, plants, tomato mosquito, wilt virus, tobacco mosaic virus, etc. Bacteria. Bacteriophage, virus attack bacteria. Internet, my friend. Collect pictures, characteristics of different microbes, prepare a chart. We will make exercise in next video.